Okay, so I think we'll start now. We've got quite a few people here now. So thank you all for coming to the Microglia seminar series. I'm Claire. Guy is not here at the moment. He will probably be here a bit later. But basically, can I just tell people to keep themselves muted throughout the presentation so that Colm is not interrupted at all. And also, if you can, to reserve bandwidth, please turn off your video cameras until the end, and we can ask questions. If you have questions during the talk, please put them in the chat function, and then I'll ask any of the questions at the end. And if there is a little bit more discussion, can have that between people. With that, I have a little bit of a bio on Colm. So Dr. Colm Cunningham is an associate professor in the School of Biochemistry and Immunology at Trinity College in Dublin, Ireland. His main research interest is to develop and study animal models to understand the impact of inflammation, in particular systemic inflammation, on brain function and neurodegeneration. Dr. Colm Cunningham trained for a PhD in neurochemistry at Trinity College, Dublin, before spending 8 years with Professor Hugh Perry at the University of Southampton, where he first described microglial priming during chronic neurodegeneration. In 2006 and 2010, the Wellcome Trust awarded him some fellowships to establish and develop the first animal models of delirium during dementia at Trinity College, Dublin. So with that, I think I'll leave the floor for Colm. And yeah, so enjoy.
Okay, thanks, Claire. Hi, everybody. Still hasn't got any less weird talking to your own screen, but I know some of you are out there, so that's great. Thanks to Claire and to Guy for the invitation. This has been a really nice series, another nice kind of COVID initiative, and so I was delighted to get the call to give you this talk today. So I'm going to talk about microglia at the interface of chronic neurodegeneration and systemic inflammation. Been very interested in both. But there's now a very clear connection between the two, and that's what I'm going to talk about. So, yeah. Okay, I always start with this in case I forget it at the end in the high stress situation of giving talks. The work that I'm going to show you has been funded largely by the Wellcome Trust and then latterly by the National Institutes of Health in the US. These people in Trinity College Dublin have really helped with the work. Sadly, all of them have moved on to other things now, but I'm going to show work from Arshad Nazmi and Ana Belén Lopez Rodriguez and Aidan Griffin and Donald Skelly in particular. And then we have some important clinical collaborators, Daniel Davis in UCL, Alistair McCulloch in Edinburgh, and Hugh Perry, of course, who I worked with for a long time, has been very influential in much of this work. Okay. So I'm going to talk about a couple of different things, but hopefully you'll see that there's some logic to the progression. I'm going to start off by talking about chronic neurodegeneration before I go on to talk a little bit more about microglial priming and what we think underpins that. And because this, I think, has deleterious consequences for secondary insults to the vulnerable brain, I'm presenting these in a string. Sorry, my advance the slides only works in one place. Okay, so this story for me starts really when there was this epidemic of bovine spongiform encephalopathy in the UK in particular. And as a result, there was money available in the UK to study this fatal brain disease and to study the role of brain inflammation in this fatal disease. And at the time, of course, we didn't even have the word neuroinflammation. That was developed subsequently. We were looking at microglia as a manifestation of CNS inflammation. And I did this in Hugh Perry's lab. So I started there on a postdoc studying a mouse model of prion disease, the ME7 model of prion disease, which came from a disease of goats, which was endemic in the flock in Scotland and Britain at the time. And this is a really nice model system. And we were studying this because it was important to try and understand the degenerative processes that are happening in prion diseases. But it also turned out to be a fantastically useful model of how the brain responds to chronic neurodegeneration. And what's nice about it is it's not just a model disease, it's a real neurodegenerative disease. It's happening as a result of exposure to these aggregated forms of the prion protein, and it develops in a very natural way. And we can look at that entire progress of neurodegeneration. So it has a really nice, robust sequence of neuronal and synaptic changes. If you look down at the bottom here, this is the presynaptic marker synaptophysin, and you can see in the normal animals you get this lovely layered staining in the hippocampus, and this is absolutely decimated by prion disease, as you can see here with the loss of staining in the hippocampus and also down here in the thalamus. And a little bit later on, you get this neurodegeneration in the hippocampus that follows the synaptic loss. You get real destruction of the CA1 pyramidal layer, and you start to see this spongiform change in the hippocampus. And that's what gives the spongiform encephalopathies their name, of course. And then important to say that there's a really robust sequence of behavioral and neurological changes that comes with this. And actually this is associated with the synaptic loss and precedes the overt neuronal loss by some months. So there's a really robust progression of disease here. And what I was paid to look at the time was the microglial response. And this is shown here just using CD68, which is an endosomal marker that many people use as a metric of phagocytosis, phagocytic activity in the microglia. So you can see here there's very robust CD68 expression. You can just about see the CA1 layer in between. So these cells are dying, they're losing presynaptic terminals and postsynaptic dendrites, and these microglia all become phagocytically active at this time. And when we look at that in a little bit more detail, this is before single cell RNA-seq, and we were looking at quantitative PCR for just lots of different things that we thought might be activated in the ME7 model. I should say MBH stands for normal brain homogenate, because this is a surgical model. So we inoculate animals with an infected brain homogenate, and that starts to develop this chronic disease that I've shown you. And the typical control group for that is to inject them with a normal brain homogenate. So you'll see this repeatedly. So when we look at the fold increase in the disease compared to the normal animals, you can see that there's a very robust induction of some of these scavenger receptors, CD68 and SRA2 in particular. Lots of lysosomal cathepsins showing a real big increase in lysosomal activity in those guys. We have some matrix metalloproteinases, but not all, very low MMP9, but very high MMP3 and MMP12. And then the UPA system is very activated, while the tissue plasminogen activator is not. We have robust activation of subunits of the NADPH oxidase, and also some other microglial markers, TREM2, which of course has become a real focus in the neuroinflammatory field recently. And here's the peripheral benzodiazepine receptor, which of course is the target for the PET ligands that are used to look at neuroinflammation non-invasively. So there's a huge panel of different microglial markers that get activated in the prion disease model. So it's a very robust neuroinflammatory response. Now, one of the things that we got interested in quite a while ago now, actually, even though this is quite a recent paper, was to look at the type 1 interferon response in those animals. So you can see here that in the ME7 animals, there's a very robust induction of interferon beta, which is one of the type 1 interferons. And if we look downstream of interferon beta, you can see IRF7, which is kind of the primary readout of action of interferon beta. This is all transcriptional work, but IRF7 is very reliably induced downstream of interferon beta action. And then the rest of these genes, these are all very robust parts of the antiviral response. So there's a whole raft of things that are induced as part of the machinery to try to block viral replication within the cell. So there's if you look at all of these in sum, you can see that there's a very robust induction of interferon beta, and all of these downstream interferon sensitive genes also get turned on. Now, this was bulk RNA, so we couldn't say which cell type was expressing these, but more recently we then isolated astrocytes and microglia from these animals to look at who might be making this type 1 interferon. And you can see there's a very robust induction here of interferon beta in the microglia, and really nothing to speak of in the astrocytes. Now, if we look downstream again at IRF7, the microglia are also responding to that interferon, and there's a very well-known autocrine action of interferon beta released from the microglia or from whichever cell makes them and acting again on the same cell. But of course, they can also act on local cells. And you can see here that the astrocytes, although they're not making type 1 interferon, they also respond to type 1 interferon by transcribing IRF7. So there's a robust type 1 interferon response. So we were wondering, of course, what it does to the microglial activation. And here we were, I suppose, initially slightly disappointed to see we inoculated IFNAR1 knockout animals with prion disease. So this is the receptor for interferon beta. You get no activation of the downstream genes in these IFNAR knockouts. So when we looked at the microglial morphology, the activation in prion disease, it was absolutely clear that the microglia get activated even in the absence of interferon signaling. And this is, you can see it very clearly here at low power and also at high power. There's a very significant change in morphology from the kind of quiescent microglial cells that are in the MBH animals. But if you start to look at the phenotype, then some of the changes start to emerge. There are a number of things that are not altered, including the morphology, as I said, but also CD11B and CD68 here at the hippocampal level weren't really changed. Getting a lot of prompts that people are entering the waiting room still. So there's changes in CD11C, ITCAX, TMEM119, and TREM2. They're significantly upregulated in disease, but suppressed in the IFNAR1 knockouts. If we look at cytokines, TGF-beta, TNF-alpha are also suppressed in the IFNAR knockouts. They're not really induced in the astrocytes in the first place, they're induced in the microglia. And interestingly, it didn't have any impact on IL-1 beta expression. And then we can look at things like the complement pathway. Both C3 and C1q are induced by disease and suppressed in the IFNAR knockout. If we look at lysosomal proteins like this cathepsin D, very robustly induced in disease and really completely suppressed again in the IFNAR knockout. And also for the SIB A and B, which are components of the NADPH oxidase, again increased by disease, suppressed in the IFNAR knockouts. So so that was interesting that there was such a striking effect on the complement pathways and on lysosomal pathways. So we had a closer look here at CD68 and cathepsin D, and particularly in the white matter around the hippocampus. We know the hippocampus is degenerating in this model. And if you look at the white matter, you can see a really robust induction. This is IBA1. And if we look down here at cathepsin D, you can see a striking cathepsin D activation in the white matter that's more or less absent in IFNAR1. Now, actually, I thought CD68 was on this picture too, and it's not, but you can take it from me that CD68 also in the white matter tracts was reduced. So it looks like there's a suppression of the phagocytic activity in these microglial cells in prion disease when IFNAR is not active. And this has a consequence for the disease, and these are the synaptic and neuronal changes that I showed you in the first slide. And you can see that prion disease decimates the presynaptic terminals in ME7, but in the IFNAR knockout, this is significantly mitigated. Now, it's not intact by any means, so there's still damage happening here, but you can see that the density of presynaptic terminals is very much reduced and protected to a significant extent in the IFNAR knockouts. And again, if we look at the neuronal level, this is with NUN, this posterior part of the thalamus here shows very significant neurodegeneration in prion disease. And again, we see some protection of that in the IFNAR knockout. And if we look at neurological changes in the disease, these are neurological tests of the time the animal can hang on a horizontal bar or indeed exit from the horizontal bar, and time on an inverted screen. And you can see these decline with prion disease, and you get some protection against that decline. Only a delay, not a prevention, but you do see some protection against that decline in the IFNAR knockouts. And that also is borne out in the mortality data. They all die, but they survive for longer. So it's having an impact on the progression of the disease, on the neurodegeneration and the neurological scores. So it has some impact, it has some contribution to disease. So we were obviously interested to go a little bit further and see how that came about, because that wasn't really clear. Lots of people were describing type 1 interferons in the brain. In fact, in lots of the models that have come out in the last few years with single cell RNA sequencing, there's a kind of little module of type 1 interferons that's coming up again and again. So we had a little bit more of a look at that. This was some in vitro work that Aisling Dunn's lab did in Trinity, and we worked with her on this. So she was isolating microglia and astrocytes to make these primary cultures and looking for a whole panel of nucleic acid sensors. Aisling is an immunologist, and these nucleic acid sensors were being described at the time in lots of different immune cells. So she looked at them in the microglia, and you can see here microglia expressing lots of P2O4, of AIM2, which is a component of the inflammasome, and CGAS, which is part of a complex that recognizes double-stranded DNA to trigger the type 1 interferon response. So all of these are expressed on primary microglia. And then if you challenge those microglia with DNA or double-stranded RNA from different sources, you can produce a really robust type 1 interferon response from these primary microglial cells. So CGAS STING is a relatively recently described system for the recognition of microbial DNA. You can see here DNA from bacteria and viruses can be recognized in the cell cytoplasm. And it also transpires more recently that DNA, self-DNA from dead cells, and even mitochondrial DNA from damaged mitochondria can also be recognized by CGAS. And so what CGAS does is it converts a guanosine triphosphate and adenosine triphosphate into a dinucleotide called 2,3-cyclic GAMP. So that's the entity that's recognized by the STING protein on the surface of the endoplasmic reticulum. And on binding this dinucleotide, it's able to activate these two different signaling pathways. It activates TBK1, which phosphorylates IRF3, and it also activates IKB kinase, which phosphorylates IKB alpha, leading to its ubiquitination and degradation, which leaves NF-kappa B free to translocate to the nucleus. So the activation of STING by these dinucleotides leads to an activation of the IRF3 pathway, which induces type 1 interferons, and also the NF-kappa B pathway, which induces obviously a whole host of inflammatory genes. So we had a look, because these were available to us locally, we had a look at the STING knockouts in the prion model to see if this would make any difference to their interferon beta responses. And this was extremely clear. These are the ME7 animals, which we've used to inoculate wild type and STING knockout animals. And you can see that the type 1 interferon response that I've shown you already is completely absent in the STING knockout. And likewise, if you can go downstream and look at IRF7, again, it's almost completely absent. So the type 1 interferons made in prion disease are being made through the activation of the STING, the CGAS STING pathway. And then just to look at some of the other things that we looked at in the IFNAR knockouts, again, we can see changes in the complement pathway are suppressed in the STING knockouts, and changes also in cathepsin D, which I've already shown you. So STING is necessary for the induction of type 1 interferon in the model of prion disease, and IFNAR1, this interferon is then acting through IFNAR1 to change the microglial phenotype and also to influence disease progression. So there's lots of things we don't know about this still. As I said to you, the microglial activation is still occurring in the absence of IFNAR1 and of STING. So they change their phenotype, presumably due to early degenerative events like detection of synaptic debris. Somewhere along the line, and we're not sure exactly when, they must be also exposed to ligands for STING, whether that's this dinucleotide cyclic GAMP or mitochondrial DNA, or even endoplasmic stress, which has also been described to activate STING. The microglia are becoming activated and producing type 1 interferon, and this can then act directly on the microglia in an autocrine way or on other local cells. And in the absence of either of STING or of type 1 interferon, lots of these microglial genes that we've seen activated in prion disease are suppressed. I've focused mostly on the lysosomal here, cathepsin D and CD68, but also there are changes in COX-1 and prostaglandin E2, changes in TREM2 and TGF-beta, the complement pathway. So lots of these things are being shown to be contributory to degeneration in these models. And IFNAR is controlling these to some extent. So it's shaping microglial phenotype, and it's also contributing to neurodegeneration. And this is emerging now, this STING pathway is emerging in models of EAE, TBI, prion disease here, and ALS, the model of motor neuron disease. So definitely one to investigate a little bit more closely. But I'm going to move on from there to talk a little bit about microglial priming, because this happened relatively early, actually, in my time with Hugh Perry in Southampton. We got interested in how systemic inflammation can impact on the degenerating brain. So this headline here is obviously not going to be news to many of you, that the brain is connected to the rest of the body. But in terms of neurodegeneration, that's not something people were really thinking about. Well, God, it's almost 2 decades ago, which is embarrassing, but we started to think about that a long time ago now. And this kind of came from the sickness behavior field, because we knew that as inflammation in the periphery is mounted to fight infection, or as a result of local injury or whatever, that these cytokines that are part of regulating and coordinating that innate immune response, they're circulating in the blood. And we learned quite a long time ago that they can signal, even across an intact blood-brain barrier, they can signal to the brain to bring about changes like the fever response and the sickness behavior response, and can also induce further cytokines and prostaglandins inside the brain. So we were simply interested in what happens to those signals when they arrive to a brain that's already inflamed because of existing neurodegeneration. And that's what we were seeing in the prion disease brain. So that's where we started to ask this second question. If we superimpose systemic inflammation on that brain, what do we see happening in the brain? And this was, as Claire mentioned, this was the description of microglial priming, which is also a very old observation now, but it's still kind of been formative, I suppose, for many of the things that I've pursued subsequently. And it's still important in this story. So these are the microglia of the normal brain. They're very small cell soma and highly ramified branches. When prion disease develops, as I've already said, they increase in number, they change their morphology. But when we look by immunohistochemistry, at least for IL-1 beta, we couldn't detect that. Now that's not to say it's not being made at all, but by these methods, we can't detect any IL-1 beta. But when you then challenge these animals systemically with bacterial LPS, their morphology doesn't look a whole lot different, but now they start to make IL-1 beta. So you can see this blood vessel here is positive for IL-1, and then all these parenchymal microglial cells around the vasculature also become positive for IL-1. And if you make exactly the same challenge to normal brain homogenate animals, that always seems to happen, then these parenchymal microglial cells don't make any IL-1 beta. So at that time, we described these microglia in prion disease as primed to produce exaggerated responses to subsequent inflammatory stimulation. And it was a functional readout. It was IL-1 beta was the switch in phenotype, that this exaggerated production of IL-1 beta was what was happening to those microglia under a secondary stimulation. Now, we and others have shown that that's not specific to prion disease. This is just one picture from our own lab, from this paper from Ana Lopez Rodriguez, showing the microglial cells in the APP-PS1 model of Alzheimer's disease. And you can see these gather around the amyloid plaques quite differently to the wild type animals here. But again, if we look for IL-1 beta in the disease per se, we don't really see it until we challenge those animals with bacterial LPS or even with IL-1 beta. And then you get these clusters of microglial cells all around the amyloid plaques. It's not all of the microglia. It's not right through the hippocampus. It's particularly in the microglial cells around the plaques. And you can see that very clearly here, IL-1 beta positive cells right around the 6E10 positive plaque. And those cells, when you double label with IBA1, shows very clearly that these are microglial cells. So again, the amyloid plaque is priming the microglia to produce exaggerated responses to subsequent stimulation. And as I say, it emerged then that a number of different disease models and aging itself produce this priming of microglia. This is adapted from a figure from Inga Holtman, and it just shows that there are lots of different, slightly different microglial phenotypes in chronic diseases and in aging, but there's a shared signature that all of these show, which shows induction of phagosome and lysosome components, more components of the antigen presentation machinery, more oxidative phosphorylation, and then a number of specific genes like AXL and CLEC7A, LGALS3, and ITGAX CD11C there, that are shared among all of these different models. So they're not all the same. The microgliosis is not the same in all of them, but there's a shared signature in all of these. And importantly, this signature here in blue is quite distinct from what microglia do when challenged with LPS, which is a much more NF-kappa B driven type of activation. So there's a clear separation of those two phenotypes. And of course, when we're challenging with LPS in the periphery, these prime microglia are now switching to this sort of phenotype, but in an exaggerated way, making more IL-1 beta. So we were interested to look at that in a different model system, because of course in Alzheimer's disease models, you have a lot of amyloid, but really very little neurodegeneration. And we became interested in what would happen to the microglia when there was degeneration of the cholinergic system, which is a major feature in Alzheimer's disease, and to an extent also in aging, that the basal forebrain cholinergic neurons start to die. And it means that the hippocampus, many other regions too, but the hippocampus starts to lose its cholinergic innervation. And we were curious to know what would happen to microglia in the hippocampus when they no longer had this cholinergic tone. So we use this toxin. It's a specific toxin for the basal forebrain cholinergic cells, because it's a ribosomal toxin linked to an antibody against the P75 neurotrophin receptor. And that's really enriched on these basal forebrain neurons. So if when you injected ICV, then you kill those neurons and you get a denervation of the hippocampus. So what we do then is we perform that surgery, inject the toxin ICV, allow the animals to recover for 35 days, and then we challenge them with LPS. And 3 hours later, we isolate the microglia to see what they've done. So this is just a picture of the model. You have the choline acetyltransferase positive neurons here in the medial septum, and you can see these are depleted by the toxin. We deliberately had a partial lesion, not a complete lesion, because nobody loses all their cholinergic neurons. And you can see the chat innervation of the hippocampus here is very much depleted in the lesioned animals. And that's associated with the clear hippocampal microglial activation, both an increase in numbers and a change in the morphology. So the cholinergic withdrawal clearly activates the microglia. And then if we look at that primed signature that I just showed you, we have increases in ITGAX or CD11C, CLEC7A, and AXL, but a decrease in this SAL1, which some of you will know as a sort of a homeostatic marker, which tends to go down as other aspects of microglial activation goes up. So it's quite an important controller of microglial phenotype, and as it's reduced, several microglial markers go up. And what's interesting is these are very, very closely correlated with the CHAT levels, the choline acetyltransferase levels in the medial septum, from which these cells project to the hippocampus. So SAL1, the homeostatic marker, is actually quite tightly regulated, or correlated rather, with the loss of CHAT in the medial septum. And all the activation markers we look at here, ITGAX, CLEC7A, and AXL, are also significantly correlated with the CHAT levels in the septum. So as the CHAT levels go down, the microglial priming markers go up. So that, of course, made a prediction that these microglia would be primed and would produce this exaggerated IL-1 functional response that we described before. So we did this by FACS staining of the CD45 positive microglia. And you can see here, if we use PE to stain for IL-1 beta, the LPS does produce a small amount of microglial IL-1 beta in normal animals, but in the lesioned animals, we get a very much exaggerated expression of IL-1 beta there, which is shown here. The frequency of IL-1 beta positive microglia is much higher in the LPS treated sapperin animals than in the PBS animals. An interesting CD11C or ITGAX, which has always been very reliably induced in any models of priming we've looked at, actually predicted very well the level of IL-1 beta expression. So IL-1 expression was very strongly correlated with CD11C in these FACS assays. So the prime microglia here are making exaggerated IL-1 beta responses. And because we have withdrawn the cholinergic innervation of the hippocampus, we examined the hypothesis actually that we could sort of restore that by giving back cholinergic agonist. So we use this, brain permeable drug PNU 282987. It's a nicotinic cholinergic agonist. So many of you, maybe all of you, will be aware of Kevin Tracy's work and the idea that macrophages have nicotinic receptors, and their anti-inflammatory receptors can suppress LPS-induced cytokines. This is in the periphery, but there was some evidence that these receptors are also present on microglia in the brain. And we looked at this idea here by lesioning the animals with P75 saprin, as I already said. It removes the cholinergic innervation of the hippocampus, but we decided in this experiment that across 5 consecutive days, we would give this nicotinic agonist to see if we could restore a more quiescent or homeostatic state of the microglia. And then just as an additional control group, we also gave this drug just a single time to normal animals that hadn't been lesioned, just to see if the cholinergic agonism was able to do anything to the LPS response per se in normal animals. So this is slightly complicated at a glance, but I hope will make sense when I explain a couple of things. So here's SAL1 first, which is a homeostatic marker, and you can see that it's high in the PBS animals. So if you haven't been lesioned, then you have good, robust SAL1 expression. And you can see by contrast, the lesion animals over here have a very much reduced SAL1. So LPS reduces SAL1, which is interesting. And the lesion that I've already told you about also suppresses SAL1. But if you then give those animals the PNU drug, the cholinergic agonist, then that could really restore SAL1 to normal levels. And the prediction then is that that might turn around the primed phenotype, and you can see very clearly here, CLEC7A is low in the normal animals, but high in the lesioned animals. And again, PNU is actually bringing those levels almost back down to the baseline levels. And the same is true down here of ITGAX, which codes for CD11C, very much elevated in the saprin lesioned animals, but returned to baseline by the cholinergic agonist. And the prediction from both of these two findings, these are part of the primed signature, is that the IL-1 response would now no longer be exaggerated when given to those lesioned animals. And you can see here, LPS in a normal animal will produce IL-1. Those levels are exaggerated when you do it in the lesioned animals. But if you do that in the lesioned animals, but have treated them prior to the LPS challenge, you've treated them with the cholinergic agonist, then this brings the IL-1 response right down. Now this agonist was not on board at the time of the LPS challenge. It's important to say that when you give the PNU drug at the time of LPS, here, it doesn't make any difference to it. So it's not simply the case that the nicotinic receptors on the microglia prevent an IL-1 response. They clearly don't, but there is a prevention of that exaggerated IL-1 beta response when the nicotinic agonist has been, the cells have been exposed to this nicotinic agonist. So there's a priming of the microglial cells by the denervation, the cholinergic denervation, but actually we can reverse that priming by presenting this cholinergic agonist, this nicotinic agonist. So we think that that might be important, that the loss of cholinergic input to the hippocampus and to other areas, of course, may leave those microglial cells primed, and also that would contribute to leaving the brain more vulnerable to a secondary insult. So we can kind of update that Holtman slide with the prion disease model I've shown you and the cholinergic degeneration model I've shown you. In both cases, we have that shared signature in here, but we're adding a little bit more about the interferon 1 story, but also that cholinergic degeneration contributes to this phenotype, and we can actually revert the phenotype back towards this surveillance type by restoring that kind of cholinergic tone. So yeah, I think that shared phenotype is very important. I think to a certain extent it's also shared in all of these new phenotypes that we're hearing about in the single cell RNA sequencing experiments of the last couple of years. Obviously there are distinctions between these different phenotypes, but just how different they are is not entirely clear, because clearly there is a certain amount of that phenotype that's shared. The focus, of course, has been on what's different between the phenotypes, but there is some commonality to them too. Okay, so so that's the cholinergic influence of microglial priming. Using the prion model, the cholinergic model, and the amyloid beta model, we clearly see that, that microglia become primed in a fairly general way. In multiple different types of degenerative models, the microglia become primed, they become vulnerable to exaggerated responses upon a secondary stimulation. So the key question for us, and actually for our lab for quite a long time, more than the microglial details, has actually been about the functional outcomes. Does it actually matter that these microglial cells become hypersensitive? So are there deleterious consequences of those secondary insults? And the focus here, again, is that these cells are making exaggerated IL-1, and does that matter for the brain? Well, first of all, does it happen? Does it happen in humans? And that's quite tricky to look at. We don't have very good data on that still. But this is from Delphine Bosch, who helped us with this part of the study that just is impressed now from Anna. And what this study was, post-mortem patients with Alzheimer's disease who died with an infection and those who died without an infection. And looking at the brain homogenates from those individuals, you can see that there is an increase in IL-1 beta in the patients who died with infection. It's hard to detect IL-1 in the brain in many of these patients, but on the whole, the levels are increased in those who died with infection. This is perhaps easier to see, the phenotypic switch to a more inflammatory state if you look at IL-6 here. But it's quite interesting. IL-6 and IL-1 are correlated in the Alzheimer's patients with infection, and they're not in those without infection. So acute inflammation, I'm showing you infection here, but this is also true for hip fracture, which is very common in elderly patients. You get an increase in brain IL-1 beta upon these acute insults. So it doesn't demonstrate microglial priming, but it does demonstrate that the inflammatory status of the brain undergoes a phenotypic switch when those vulnerable individuals are exposed to a secondary inflammatory stimulus that is occurring outside the brain. The other really good evidence that systemic inflammation, acute systemic inflammation, has bad outcomes in humans is delirium. So delirium is wildly common neuropsychiatric problem that we see. You know, I mean, there's up to 20% of patients in a 500-bed hospital experiencing delirium around now, and this is even worse at the moment because of COVID, because really there's a very large incidence of delirium in patients who are COVID positive. So delirium is a really profound, it's acute and transient impairment of consciousness. So patients' thinking and memory and attention become profoundly impaired. They have changes in their psychomotor behavior. So some become hypoactive, some become hyperactive, and many of them have hallucinations, very fearful experiences. So there's a lot of perceptual and emotional changes, and it's really very distressing for these patients. But for many years it was sort of written off as, you know, the patient is confused, it'll pass, and to some extent it's true that it will pass, but it's become clear over the last decade or two that this also leads to an acceleration of underlying dementia. It shortens times to new institutionalization, it shortens times to death. So there's a lot of negative sequelae to episodes of delirium. And apart from anything else, it costs billions in extending hospital stays of patients. So it's really quite a big problem. And the pathophysiology is very poorly understood because it's largely been neglected over the years. So we don't really know how it works, but we do know that the key risk factors include aging and dementia, and the key acute triggers are systemic inflammation and many other things, but that's present in a lot of cases in which delirium is triggered. Infections and traumas and surgeries can contribute to episodes of delirium. So we thought we might try to model that using the sorts of systems that I've just shown you. We've actually done it in all three systems, but I'm just going to show you data from the ME7 prion disease model. And we simply asked the question, what would happen to acute cognitive function when normal animals and diseased animals, normal animals and diseased animals were challenged with bacterial LPS. And I'm not going to go into this maze today because we have a little bit of a shortage of time, I suppose. But what I will tell you is that to successfully solve the maze, to successfully alternate in the maze, you have to pay attention to where you first visited in the maze. And then when you're put back in this maze 25 seconds later, after being in a home cage, then you're the mouse is allowed to choose either direction, but must learn to alternate from its first choice to go to the other side, again, to find this exit, to exit the maze. So this is a shallow water maze because we have to motivate LPS treated animals to move, and they don't really do anything when they're feeling sick. Got some conversation going on there. So anyway, this is alternation is the assay here. So that's what we're measuring on the y-axis. And a normal animal would typically have about 80–90% alternation in this maze. So this is the day before challenge with LPS, and they're doing fine. Whether they're diseased or not, they're doing fine at this time. But we can unmask a sort of a vulnerability by challenging those animals with LPS. And now you see an acute working memory dysfunction that lasts for several hours. But that LPS challenge is only doing that in the ME7 model. So you have equivalent systemic inflammation in normal animals that doesn't produce any cognitive deficits, but there's a reduced and very robust acute and transient working memory deficit. So this was a little bit, or seemed resonant with the episodes of delirium that we see in these elderly patients. And we looked a little bit further at this as the underlying prion disease progressed. We were able to show that the same sorts of challenges could now produce much more severe deficits. We could show that these deficits were fluctuating, which is a feature of delirium, and that they lasted longer. So this made a prediction for humans that the underlying progression of disease would progressively increase the risk for delirium. And this epidemiological cohort that we studied, the Vanta 85 cohort in Finland with Daniel Davis, showed that as baseline cognition gets lower, the probability of delirium gets higher. And that's exactly what we showed here, albeit with an animal model where we really can't say these mice truly are delirious. That's not an easy thing to do in a non-verbal animal. But as disease progresses, the brain becomes more and more vulnerable to the impacts of a systemic inflammatory challenge. Now, not only does the brain get more vulnerable as underlying disease progresses, but actually episodes of delirium can also change that rate of decline. So I mentioned this to you before at the introduction, but these, again, this is the Vanta 85 study. And if you follow these people up over several years, you can see that the mini mental state examination goes down quicker. Their scores decline more quickly when they've had an episode of delirium than when they haven't. So even if these patients, these people in the community reach a diagnosis of dementia, the decline will be more rapid if they've experienced episodes of delirium. So it suggests that there's some de novo pathology or injury that's occurring during the delirious episode. It doesn't seem to be related to increased levels of amyloid beta or tau. So it does suggest that there's some sort of de novo event. And we actually, earlier than that data came out, we had already shown something like this again in the prion disease model, that if you look at the progression of prion disease, then a single challenge with LPS back here, before this graph even starts, a single challenge with LPS was able to change and deflect that progression of disease. So these are the diseased animals that didn't have LPS, and those that were challenged just a single time with LPS declined more rapidly. And we had shown in this study, it's actually the same paper as the microglial priming study, that actually there was more apoptosis in these ME7 LPS animals compared to ME7, the disease without the LPS challenge. And those neurons, using caspase-3 here and neurofilament, were able to show that there was new apoptosis of neurons. So LPS challenge is killing neurons in the brain. It's changing the trajectory of subsequent disease. And when we, much more recently, when we did those same experiments in IL-1 receptor 1 knockout mice, you could see that the increase in apoptosis that's triggered by LPS is actually mitigated in the IL-1 receptor 1 knockout mice. So it suggests that IL-1 has some contribution to this new neuronal death events happening in those individuals. In this case, mice, obviously. So also in that same study, we looked at the vulnerability or the sensitivity of neurons in the prion disease brain to acutely applied IL-1 beta. So again, you have MBH animals here and ME7, and you can see that at 0.1 nanogram per mil dose of IL-1 really does very little to the resting membrane potential of neurons. These are CA1 or hippocampal slices from normal animals. You can see IL-1 is not really doing anything to the resting membrane potential. If you take slices from ME7 animals and apply IL-1 beta, they start to depolarize slightly, and when they go above a certain threshold, then they start to spike. This is a form of non-synaptic depolarization, but they spike really ferociously across this period of time. So this is something that they're highly susceptible to in the prion disease animals compared to normal animals. It's IL-1 receptor 1 dependent. As you can see here, it doesn't occur in those with the knockout of the receptor. And in fact, in these animals, as they have this depolarization, this actually doesn't wash out. So they don't recover their normal resting membrane potential after this spiking event. And so, of course, what that suggests is these cease to be useful neurons. They can't return to normal resting membrane potential. So this is not actually demonstrating neuronal death in this case, but certainly a sort of lasting neuronal dysfunction. So IL-1, these animals are producing exaggerated levels of IL-1, but in turn, the neurons are also hypersensitive to IL-1 beta. So it is suggestive of a role for IL-1 beta in neuronal death and also in dysfunction. And the T-maze deficits themselves are also contributed to by IL-1. This is IL-1 receptor 1 antagonist, which suppresses, not completely, suppresses the LPS-induced impairment or disruption of the T-maze alternation, the working memory task I showed you. And we can replace LPS with IL-1, so IL-1 on its own is sufficient to produce a similar sort of dysfunction. And again, the vulnerability is important because if you give IL-1 to normal animals, you don't get that dysfunction. So there's a susceptibility there that makes them hypersensitive to multiple different stimuli, but among them, LPS and IL-1 beta. So, so systemic inflammation, we have systemic or peripheral immune cells like macrophages and neutrophils are able to produce inflammatory mediators like IL-1 beta, TNF-alpha, IL-6, but also prostaglandins. And they can activate the brain by a number of different routes. I'm only showing the brain endothelium here. The endothelium can become activated, can secrete levels of prostaglandins again, but also can secrete IL-1 beta and TNF. And these can act obviously on local inflammatory cells. If the microglia have become primed by these aging or neurodegenerative changes, then they show exaggerated production of cytokines, particularly IL-1 I've shown you, and they can also produce more nitric oxide and reactive oxygen species. So there are a number of different mediators that we know can have direct impacts on neurons, and I've shown you a couple of direct impacts on neurons here. I haven't had time to talk about this at all, but these cytokines can also directly act on local primed astrocytes, producing lots of chemokines, which can also feed back and contribute to the activation and infiltration of monocytes and other leukocytes. So systemic inflammation can have multiple impacts on the brain. These are particularly severe if the brain is already activated by existing neurodegeneration. And I'm going to finish on this slide. It's particular really to this study that's now in press that I mentioned, but I suppose we can make it a little bit more general and use it as a summary slide here. I've shown you that when you have synaptic loss and amyloid beta and also cholinergic loss, these can lead to a progressive microgliosis. I think we all know that now and accept it. But here I've shown you a little bit about the activation of the CGAS STING pathway to produce type 1 interferon. I think that contributes to the phagocytic and obviously destructive microglial phenotype. So by blocking this pathway, we're able to suppress to some extent the neuronal damage in that model. And then obviously the denervation that I showed you, the cholinergic denervation, we were able to reverse that using a cholinergic agonist and actually reversing that primed phenotype, bringing it back to a homeostatic phenotype. And then I think all of this is relevant to the baseline state, the baseline neuroinflammatory state in these aging populations, particularly the degenerating populations, but it's true also in aging to an extent, such that they then become more susceptible to brain sequelae of infection. and in this APP model, but also in the other models I showed you, and we get an exaggerated production of IL-1 beta, which may contribute to some of the downstream problems. I showed you some acute cognitive dysfunction, some acute neuronal dysfunction, and also some acute brain injury, neuronal death. And the proposal is that this leads to an accelerated trajectory, which I've shown you occurs in patients, but also in the prion disease model, I showed you that they decline more rapidly. So I think I'm going to finish there, and I hope that this has been of some interest to some of you, and obviously I'm happy to take some questions.
That's great. Thank you, Colm.